Last time we talked about how the skull aberrates the beam, and when we're talking about, what does the focal spot look like across the skull, it'd be really good to have a good model to do that, or a good way to do some modeling. So I thought we would talk today about modeling. It's just going to be a start on modeling because it's kind of a big topic. All right, what do you think, Keith?
It sounds good. Kim, are you wearing the same clothes as the last lecture? Oh, wait a minute. I am too. What are the
I
odds
cannot
of that?
believe you asked me that. Let's talk about ultrasound. All right, so first of all, I want to go over sort of the big picture on how the modeling works before we get into the details. And we're going to go through it part by part, but let's just talk about the big picture first. If you're going to do a simple model of the focal spot in water, this is really easy. And this is something that students are going to be working on in their MATLAB with some MATLAB code right away. And the basic idea is that for every pixel in the medium, you're going to find the contribution of one particular element in that point in time. So at this point in time, there's no contribution, no pressure contribution to where my mouse is, but a little bit later when the wavefront has come out here, then there is going to be a contribution to it from that element. So it's going to store that value for that point in time, and then it's going to do this for all points in time, and then for all elements, and all points in time, all points in space, and then it's going to do it for each of the elements. And when it sums up multiple elements, then you're going to start to see the constructive interference and destructive interference. And then once you have a pressure wave and you've got the summation for all the elements over all points in time, then you can just say, well, what's the maximum that was reached at all points in time? And then that can get you the maximum pressure. And then you convert that to intensity by squaring it and dividing by Z. Does that make sense to you, Keith?
It does. And it also tells me why it probably takes so long to run some of these models, especially if you're doing a lot of layers and voxels instead of pixels.
Yeah, now this one is going to be relatively fast, the one that we're going to give students to work on, because it's just going to be 2D simulations of just one plane, it's not a whole 3D. And it's just there's a lot of things it's not doing. So it's really just such a simple calculation that it's only going to take a few seconds to come up with an image that looks like this. And these are important images. There's lots of times you just want to know, what does the beam look like? And so if you are thinking about what kind of ultrasound transducer do you want, what are your trade-offs with the pitch and the frequency and the depth, the radius of curvature, this is a great way to do that.
So people would use these models before they've designed a transducer, and would they also use them once they have a transducer and a product built?
So the thing is that once you have your transducer, sure, you can do this and you can say, this is what your beam looks like in a water bath. The thing that is now going to get really complicated is that if you're going to do transcranial ultrasound, is that the skull is going to aberrate the beam and it's not going to look like that. And then you have to do a much more complicated simulation. I wanted us to talk about the elements of that. I don't know how far we're going to get in terms of the homeworks and in terms of students doing full beam simulations, but there are certainly elements of this that I want the students to work on. So let's talk about the elements, because I want everybody to get sort of on the same page as what are the elements before we talk about, go down and talk about each one individually. So the first element is modeling the transducer. So we've talked about that because Huygens principle says you can model the transducer, even if it's a single element or multiple elements, as individual points. And then you can sum up the contribution of all those individual points. You have to know what your configuration is, whether it's a flat transducer or whether it's a curved transducer. There's going to be components for like the frequency, the pulse length, and things like that. So those are things that are going to be elements of the simple MATLAB code of the prior slide, as well as this one. So these are things that we've talked about, and people should have a good idea of what I'm talking about those right now. The other thing that we've talked about is calculating intensity. So if you can calculate the pressure wave, like we've mentioned in the prior slide, then to get to intensity, you take the square of the pressure and divide by Z. So calculating intensity is relatively straightforward. It's the rest of it that we haven't really talked about. So let's just go back up to the beginning. You've modeled your transducer and your element configuration, and there's individual points, and you're going to do a beam simulation. First of all, the next thing you have to do is you have to say, well, what are the acoustic properties for every pixel in your volume? And in order to get that, maybe you have an image, like a CT image or an MRI picture, such as the one shown here. So this is a CT picture, and it's really just showing you the skull. And now you can say for your 3D volume, you can assign acoustic parameters based on that image. So we'll have a whole discussion now talking about how acoustic parameters based on CT or MR. And so acoustic parameters, of course, we're talking about speed of sound and we're talking about attenuation. So for every voxel, we're going to have to assign those acoustic parameters. If we have an array, we might then apply our phase aberration correction terms. So I'll talk about how do you do that in a simple method. So then we might say, well, we're going to, if it's an array, this is of course our single element transducer, but let's pretend it was an array. And then you would calculate what those terms would be, and then you'd apply those, and then you'd do the beam simulation again. And then to do the beam simulation, you propagate the wave. And so it goes some small time increment. It's going to go some small distance. And as it reaches into each new voxel, there's going to be attenuation. There's also going to be reflection, transmission, refraction. You've got to calculate the refraction term as well. And so it goes voxel by voxel as it calculates that. And then it can sum up all the wavefront at the focus. So it's a little bit more complicated than what I just described in the prior slide. Quite a bit more to do there. And then at the end, once you've got your intensity, maybe then you're interested in the temperature rise, and then you calculate the temperature from the intensity.
And all of the simulations the students are doing are two-dimensional, is that right?
Well, for the MATLAB code that we're going to give them that is based on the prior slide, it's just a 2D simulation, yeah.
But if you wanted to do it in 3D, you're just kind of adding the other plane and doing more iterations, but it's still the same principle.
Yeah, yeah. To do it properly, you'd want to do it in 3D because you can imagine that there's a beam that comes out and it refracts at the surface and it comes off at an angle, or maybe it goes the refraction causes it to go through plane, like into the plane or out of the plane. So you'd want to take all of that into account. So a full 3D simulation would make more sense to do it properly. In the prior example, we didn't have any refraction. There was nothing in that water bath that was going to cause any refraction or any reflection. So we didn't have to worry about any of that. So it made sense to just kind of do the 2D simulation.
And for the phase aberration correction terms, you were telling everyone that you can model a single element transducer according to Huygens principle where there's individual, wavelet generating parts. So why wouldn't you need phase aberration correction for a single element?
Well, you don't. What you would do is assume that all of the elements have the same phase, because in reality, that's what would happen. They would, you would model it as individual points across the shape. If it's a curved, spherical shape, then you would model it as individual points, but they would all have the same phase because your single element transducer would have.
Oh, okay. Sorry. Now I'm seeing that there's correction. So what you mean is that for a single element transducer, there will be aberration that we cannot correct for.
That's right.
Oh, sorry. Okay. I understand.
That's right. Absolutely right. So having the different points there and simulating the different points allows you to figure out what the aberration is. It's something that you won't be able to correct. Yeah. Okay. So I thought the first thing we would do is talk about the temperature component. We talked in the last lecture a lot about temperature. So students know that there is going to be a temperature rise. If we use small intensities, it could be very, very small. If we use higher intensities, it'll be bigger. But let's figure out how to model that. All right, so let's do the temperature part. And so first of all, I have here a little movie of a focal spot in a phantom. So this is an MRI picture, first of all. MRI is going to give you some signal where there's water protons. This is a phantom that is a gel, so there's a fair amount of water protons in there. Outside of that is air, no water protons in air, that's just giving you noise. I don't want to get into the details of how we got the temperature map. It's just, very briefly in case anybody wants to know, it's the resonant frequency of the MR signal changes with temperature, and we can be sensitive to that. But what we see right here is the bright spot at the focus. So I'm not exactly sure whether the transducer is, I think it's above, right up here. And I think it's coming down. And so anyway, you can see the long axis of the focus. And you can see that initially the temperature is going to go up and then it's going to go down again. Now there's a number of things I want you to notice here. One of the things I want you to notice is that, that as we look over time, and we just find one time point such as this one, that we can see that the temperature rises in the same pattern as the beam plots that we calculated before. So on the far side of the focal spot, you see that there's these two parts right out here, and those showed up in our beam pattern that we calculated in our MATLAB code as well. You can see just barely make out there's a little bit brighter in the near field as well. And so that's kind of nice to see that in reality, you can make measurements that are exactly like what we simulated. The other thing, so we notice is that the temperature is going to go up in the focal spot when the ultrasound is on, and then it's going to go down again. So the ultrasound's on, the temperature rises, and then it goes down again. The time constant for the rise and the decay aren't exactly the same, but that doesn't matter right now. What we see is that that happens at the peak, but it's really interesting to me that what happens over time. If we make a plot across here, across the focal spot over time, then we get multiple line plots, as you see over here, and these are going to follow the rainbow. So the first one's red, orange, yellow, green, blue, indigo, violet. So initially what we see is that the center part goes up, and so it's very peaked. The temperature is very peaked and very narrow. And then it goes up to its highest point. As it comes down again, it gets broader and broader. And so the last one is sort of like this pinkish color, and that's sort of the broadest one of all, which is really interesting to me, because what's happening is that that's sort of outside of where the ultrasound is hitting it. And so out here, and what's happening is that the heat is moving out. So in fact, on my next one, what I'm going to do is I'm going to plot 3 different points over time. One's right in the center of the focal spot, and then 2 that are adjacent to it. What we see is in the center of the focal spot, of course, it's the most peaked when the ultrasound's on, and then it decays away. Adjacent to that, you see that it's peaked at a later time point, and then adjacent to that, it's peaked at the very end. So what's happening is that the ultrasound is depositing heat in the center, and then it's conducting away. So this is kind of the basis for one component of the heat transfer. Whenever there's a spatial gradient of temperature within a medium, energy in the form of heat flows from the region of higher temperature to the region of lower temperature. So you're putting in the temperature rise, the heat right at the focus, and then it's going to be moving away. All right, so in terms of heat transfer, our equation is going to look something like this. The heat change in any particular pixel that you're looking at is equal to the heat deposition. If you're looking at the focus, then this is going to be due to the focus ultrasound. If you're outside of the focus, then there isn't any heat deposition, and there's only these other components. So any particular pixel, you're going to have the heat deposition in that pixel, plus there's going to be a conduction term plus a convection term. So let me talk about each one of these individually. So first of all, there's the heat change. So we're going to put heat in. So there's going to be a temperature change over time. And in order to get to heat, which is a different quantity from temperature, we multiply times the specific heat of the tissue. So that's just the ratio, the amount of heat energy transferred to an object and the resulting increase in temperature of the object. So this overall component here is going to give us the overall heat and then how that's changing over time. But we're particularly interested in the temperature change, so that we separated out the specific heat there. All right, so that's going to be equal to the heat that we put in. Now, the heat that we put in is the absorbed power per unit volume. So that's our absorption coefficient times our intensity. And so that's now the heat that we put in. And then now we're going to have a conduction term. Conduction is what we mentioned before. This is what was happening in a phantom. This occurs when there's a gradient in temperature within the tissue itself that drives the heat flow within the tissue or the phantom, right? So there was a gradient in the temperature and the heat flowed from higher temperature to lower temperature. That's what we saw. And so that's what we see here. So there's a constant, it's the thermal conductivity of the tissue. And then there's the spatial gradient. That's what this is doing right here is taking the spatial gradient of the temperature over space. That's the conduction term. So that's what we saw as the heat was conducting out. And then lastly is the convection term. Now, convection is what happens when there's either air currents or there's fluid currents, and it's different than conduction. Conduction is when molecules give heat to other molecules. They give energy to the adjacent molecules. And so it's just sort of molecule to molecule interaction. Convection is different. That's where you have air currents that are moving by and cooling it, or fluid currents. Now, in vivo, the convection term is perfusion. We all know that tissues are perfused, there's blood flow, and that's going to take the heat away. So that's it's now converted into a perfusion term. We don't have air, and so we just have the blood perfusion. And this we didn't see in the phantom. Phantom was just a gel sitting there. All right, so in terms of the heat transfer equation, now this last convection term is the energy loss due to perfusion. And so this is where the heat equation turns into the Penny's bioheat equation, because Penny's is a researcher who did some work on this, and he said, well, this component back here has made up of these components. So there's the temperature difference between the tissue and the blood. That governs how much heat is taken away with the perfusion term. And of course, you can imagine if the blood is much cooler than the tissue, it's going to take away more heat. Then there's a constant there. It's the specific heat of the blood. So that's how much heat that the blood can hold. And then there's a constant. So that's the component over here that's due to the perfusion. All right, so that's what we have. Just a couple more comments about the perfusion term. We know that blood flows through capillaries. Tumors have high perfusion. And so usually with tissue, we can kind of estimate what the perfusion value is, although we don't always know exactly what it is, and we could be a little bit off. Like I said, tumors might have high perfusion, so we could be a little bit off in tumors.
So both of those conduction and convection are loss terms. So by adding them, you are losing heat from those positions.
Those are loss terms. That's right.
So I'm wondering if you're looking at a voxel that's outside of the ultrasound field, and you have no intensity deposited or no pressure deposited, even though it's getting conduction from the area that does have heat, it'll come up as 0, right?
So when I said as a loss term, that wasn't exactly right, because it sort of depends on if it's higher or lower than the adjacent voxels. So if it's higher than adjacent voxel, it's a loss. If it's lower than the adjacent voxels, then it's going to gain.
Okay, so that term depends on the surrounding area. Okay.
Yeah, absolutely, which is why it has the spatial gradient of the temperature map as part of it.
Okay. And when you run sorry for all these questions, when you run this model, do you have to start from the highest intensity and then work outwards, or is it order independent?
I think it's order independent, because you can figure out what the spatial gradient is and each of these terms, and then you can apply all of those. It doesn't matter what order you put it in.
Okay.
You would end up, what you would do is this would tell you the temperature rise over a small amount of time. So what you do is you'd say over a small amount of time, what's the temperature rise? And then you do it again And the second time you do it, now the spatial map will be different. So this is an iterative process over multiple small time points. And then each time you're going to just accumulate the additional temperature, or, you know, what the temperature changes. So we were talking about why we may get the perfusion term wrong. Tumors may have high perfusion. Another thing is that once you cause a heat rise, then that could actually cause some vasodilation, and that will change the perfusion. So there's always a little bit of uncertainty about exactly what the perfusion term is. So it affects us because if you put your heat in, and then there is perfusion, it's going to lower the temperature, the ultimate temperature. It decreases predictability just because there is that variability in the perfusion term. It will increase if the time that's needed in order to get to a particular temperature. So let's say the physician is trying to, in the case of the last lecture where we saw the uterine fibroid, he wants to treat the uterine fibroid, but there's a lot of perfusion, then he's going to have to increase how much time that he applies the ultrasound in order to get the same temperature rise. And then therefore that could actually end up affecting the near field dose. So if you have to put more energy in, then you're going to get more energy in the near field, which maybe doesn't have more perfusion. And so that, you know, that variability in perfusion could affect your near field dose. You know, this could affect the brain as well. Let's say you wanted to do a neuromodulation deep in the brain, and there's a change in perfusion that's going to affect exactly what the intensity is. We don't really have any good way of knowing what that is. And so it's going to give you a little bit of uncertainty in terms of how much intensity gets to the focal point. Okay, so it challenges, of course, we're going to assume uniform heating, assumes isotropy of perfusion-related energy losses, ignoring the presence of large blood vessels. A large blood vessel is going to take, you know, be a huge heat sink. So it ignores all of those. But generally, this kind of works. All right, should we keep going? Are there any more questions, Keith?
Nope, I'm good.
All right, so we did a study, and I'm going to tell you about the results of the study. This is some work that my graduate student, Steve Young, worked on, and then he published this just recently. And these are the parameters that he used. If you wanted to find out where he found those particular parameters, you could check out his paper. I'm going to tell you a little bit more about his paper, but I just kind of wanted to report on those in case you were kind of wondering, you know, what are the values? All right, one of the things that he did was he wanted to do a simulation of the temperature in the brain when we were doing these essential tremor treatments. And then he wanted to compare them to MR measurements. And so one of the things that he did with it first was he said, well, let's do our Penny's bioheat equation. And for a particular intensity in the brain, how sensitive are we to the conductivity parameters that we're assuming? And what he found is that he assumed, you know, he looked in the literature, came up with a number of different, like a range for the conductivity parameters. And so, well, let's plug in the highest one I found in the literature, the lowest one I found in the literature, plug those into the simulation. And this is what he found is that initially the simulated temperature is exactly the same, didn't depend on what those parameters. When we got to 10 seconds out, there was a little bit of difference. And then during that cooling period, there was also a little bit of difference. So the take-home point here is, yes, the conductivity affects the simulation, but if you use that range of parameters for the conductivity that we find in the literature, it doesn't really affect it a whole lot. So that was one take-home point. And the other take-home point is that if you look at very short time frames, like 1 to 3 seconds, it really isn't making any difference at all to have those variable conductivity terms. So that was another interesting take-home point. The other one is to look at the perfusion term. And here in the perfusion, he varied the perfusion parameter by a factor of 2. And when he did that, again, he found that the simulated temperature wasn't very sensitive to that variation. So initially, over the first 1 to 3 seconds, there's almost no variation in his simulated temperature. A little bit by the time he got to 10 seconds, and then out by 20 seconds, there was a little bit as well. So what I'm going to show you is that his simulations actually had big variation in the temperature that he was simulating, but it wasn't because of the variation in conductivity and perfusion. So we felt pretty confident that the conductivity and perfusion components of the bioheat equation were reasonably good, that they weren't sensitive to the errors that we might be putting into the system. And so what that meant is that if there were errors in the temperature rise, and again, remember we had simulation versus MR temperature rise, that the errors had to be due to the first term, which is the intensity that actually reached the focal spot. Before I get to that, let me just back up a moment and reiterate something that we just saw, which is that it's generally accepted that if you are doing a very short temperature, you're looking over a short time frame, you're looking over about 1 to 3 seconds, then you can ignore the perfusion and the conduction and perfusion terms. If you remember back to the phantom image, that initially that temperature rise went very quickly at the focus, and then over the next 10 seconds or so, it conducted out. So if you're just looking over a very short time frame, you can kind of ignore those other two components. And what that means is then that this equation is much simpler. You could, for example, if you knew what the intensity was, you could set up a phantom experiments, for example, or even in Vivo, if you knew what the intensity was, you knew what your absorption was, if you can measure your temperature rise, then you might be able to back out what is the other constant here, the specific heat of the tissue. So this actually provides an ability to kind of evaluate your experimental setup a little bit.
Kim, is it also safe to assume that the perfusion error is proportional to the absolute change in temperature, in which case we could probably ignore it for neuromodulation?
Yeah, it's going to be proportional to the change in temperature, right? So if you're looking at temperature changes that are 1 or 2 degree, I mean, hopefully not, but let's say you were 1 or 2 degrees and you're looking at an error that was, just like 10%, then you're talking about, a 1/10 of a degree. So definitely something that we can think about not paying a lot of attention to, and especially over short time periods. Okay, so now that we've said a lot about temperature and modeling temperature, and remember how in the last lecture we talked about ISPTA, the temporal average, is what's most relevant to the temperature rise. I just kind of wanted to go back to a thought experiment. And the thought experiment is when we have pulsed ultrasound waveforms versus a continuous waveform. Because sometimes I hear, people kind of say, well, if you do pulsed ultrasound, then there's not going to be as much heat as if there's continuous ultrasound. And so I often wonder, what exactly do they mean by that? And so I've set up the slide to ask this question. So, are we going to get the same heating when there's pulsed ultrasound as continuous ultrasound? So in this situation where they have the same peak pressure, the same peak intensity, then in the situation where you have now continuous ultrasound, continuous wave, you're going to have twice the ultrasound. So ISPTA is 2 times as much. Clearly, you're going to have twice as much heating in the second situation. But on the other hand, if you've set it up so your ISPTA is the same by having half the amplitude with the CW versus the pulsed wave, then doesn't it seem likely you're going to have the same heating?
It does. I'm sort of getting the feeling that you're saying all those statements about pulsing are just not true. Is that right?
I think what I'm trying to say is that you have to be careful as to what are you keeping constant.
Right.
And if you're keeping the temporal peak constant, they're not going to be the same. If you keep the temporal average constant, then they might be the same. So I asked Steve to do this, Steve Leung. So he did a simulation for me, and this is what he came up with. So this is a pulsed waveform, and of course, when you do these simulations, you have to split it up into different, very, very small time points. So he had lots of little time points. He had the CW in blue and the pulsed in the orange color. And what you see is that when the ultrasound is on, the pulse temperature goes way up, and then it seems to drop off a little bit when the ultrasound is turned off. then it goes up and drops off. And here what he did was he kept the temporal average the same in the two situations. So that means that the temporal peak was bigger with the orange one by 10/3, because this was a 30% duty cycle. So if the peak is bigger, then while it's on, it's going to go up hotter, and then it's going to cool off. And so that's why with the pulsed waveform, you get this sort of jagged appearance. But ultimately, if you take a step back, you're pretty much getting the same heating for the same temporal average. So I think that makes sense. And so there we go. That makes sense.
Yeah, I think the modeling will make sense to everyone, and you've said this already, but the equation tells you how much heat is being added or how much change you get over an amount of time. And the reason that you have to cut it up and do it again and again and again is because you need to look at the initial temperature as well, which is different at every phase here, right? Especially if you're turning something on and off.
That's right. You need to split it up into little time points, because over a little time point, there's going to be a different spatial extent of the heat. And that's now your input to the next time point. Yeah. Okay. Well, that may be the end of the temperature part. Yep. That's what I wanted to say for calculating temperature. So students will have some MATLAB code they'll be able to work with in order to simulate temperature for different pulsed waveforms. I thought at this point we would go back up a step, because what I'm doing now is getting to the more complicated part. So we've kind of gone through this from simple to complicated, and not in sort of time order of what you might do when you're doing a simulation. I hope that's okay. Part of the reason is because you can do the simulation of the temperature part independently of all the other parts, just assuming that you know what the intensity is at the focus. Then you can do the temperature simulation. But if we want to know how do you get to the intensity of the focus, now we have to do the complicated part. Okay, you ready, Keith?
I'm ready.
All right. So remember, we have our transducer. We've split it up into multiple different elements, and then we're going to propagate the wavefronts through each and every voxel in our volume. And then in every voxel, we're going to have to assign acoustic parameters. And so we're going to need our speed of sound, and we're going to need our attenuation. So let's go through that. We're also going to need...
Sorry, is it evident in your code how you should split up a single element? Is there a rule based on wavelength? If I have a 3 millimeter transducer face, how many times do I split it up for a given frequency? It's in your code.
I don't know if there's a rule of thumb. You're definitely going to get a better resolution the finer your grid size in your simulation, but
it may just be a resolution thing. If your image looks blurry, split it up more, you would say, right?
Yeah.
And reduce the voxel. Okay.
Yeah, and of course you're going to be trading off time to do the simulation.
Right, right.
Okay, well, let's go to the next part. And I've already introduced some of this for the acoustic parameters. So this part should go relatively quickly because we've talked about it already, but I did want to kind of review it anyway. So first of all, we're going to need to know what the speed of sound is for every voxel. And all the information you have is you're going to have a picture, a CT picture, or an MR picture. So what we did in order to figure out how to assign the speed of sound was we looked at a bunch of different fragments, and now everybody knows what a hydrophone is and what the scan tank experiment looked like. You put a skull fragment in and use the transducer, send out a pulse, took the skull fragment out, use the transducer, send out another pulse, measured both of those and looked at them. And what he did was he said, well, let's make a comparison. We know this was a short pulse and that there's going to be some number of zero crossings of that pulse. So we'll look at the first zero crossing and where that was different with and without the bone. And then that's the additional, that difference was additional time it took to cross the bone as compared to the water when it didn't have the bone. And then from that, he could back out what the speed of sound was in that fragment of bone. Each of the fragments was relatively small and homogeneous, so we just assumed that you could assign one speed of sound to that whole fragment. It didn't have different layers and it wasn't different on one side versus the other side. And so that one measurement was going to be good for that fragment. We did have our images, so we had a lot of CT images with different parameters, and then we could correlate what we measured for our Hounsfield units with each of those fragments with what we measured for the speed of sound. And these are some example data that we got. So the velocity or the speed of sound as a function of Hounsfield units. And this is split out for different CT parameters. We don't need to get into the details of that. We'll just use the green one because it's the easiest one to see. And basically what we were able to do is to fit a line to that, or is a little bit of a curve, not a straight line. And so that we could see as the velocity is increasing in the denser material, the denser bone, which had higher Hounsfield units. Remember, there's always that confusion about whether the speed of sound is related to linear with density. It's actually 1 over the density, whereas it's bulk modulus over the density, square root. But anyway, bulk modulus went up as well in this case, because the bone is very stiff. It has a resistance to compression. All right, well, the key point here is we have a Hounsfield units this way, and in your skull, you have a whole range of Hounsfield units. And so we could say, well, what's the velocity? You know, once we find a calibration curve like this, then we could say, well, given a Hounsfield unit, now we're going to assign a acoustic velocity to that pixel. So that's the general idea with getting curves like this. You know, one of the problems with our data was that there was still a lot of variation. And it, you know, I don't want to get into the details of that. We've been able to track down that some of that variation was due to the fact that the pores were shaped differently in the different skulls. So it's really interesting. In the future, we have to do some research in order to figure out how we can get a better measure of the pore shape, which is actually sometimes subvoxel. And so it's not clear how to do that. But if we could do that, then we would be able to have better speed of sound curves. All right, so this was just how we measured our speed of sound. And then using these relationships now, we can assign the speed of sound to every voxel based on our CT Hounsfield units.
So Kim, it looks like from your graph or from the plot that you'll have more accurate measurements with more dense skulls. Is that correct?
Are you saying there's less variation up here than there is down here?
I was going to say there's less variation to the left along those axes. Oh, I guess I can see your R squared values. It's pretty much the same for all of them.
Yeah, the R squared value is the same. I mean, I think your statement is correct, but the reason why I think it's correct is because it's the lower Hounsfield units that are from the trabecular bone that have the pores in them, and the pores being different shapes give rise to different acoustic velocities. And the higher Hounsfield units has just less pores. So the higher Hounsfield units is more likely to fall on the line with less variation than the lower Hounsfield units. It's kind of a detail that I didn't, you know, it's kind of beyond what we wanted to talk about, but it's something that my lab works on. So I'm like really into it. So it's kind of fun. All right, so that's the speed of sound. And then there's the attenuation. And the purpose of this plot is to say that unlike some of the parameters that we looked at before, the simulation is going to be very sensitive to attenuation. So in this one, we did a simulation across the skull. This is Steve's work again in that same paper, where his simulation, he said, well, let's say we, for example, assumed the attenuation was 1 Nep per centimeter versus 2 Neps per centimeter in the same skull. So it's varied by a factor of 2. And then what you see is that the simulated temperature, so this is has the same MR temperature rise, but when you varied the attenuation by a factor of 2, that your simulated temperature varied by a factor of 4. So what that meant is that we're very sensitive to the attenuation parameters. We're going to have to try really hard to get the attenuation of the skull understood as a function of Hounsfield units and to understand that as best as possible, because the variations that we're going to see are going to give us, you know, big variability in our simulated temperatures. You know, here I'm talking about temperature, and the reason why I'm talking about temperature so much is because it's such a nice data set that we have from the essential tremor. We have a temperature rise in human skulls that we can use for truth and compare it to our simulation. Now in neuromodulation, we're not gonna have a temperature rise, but what this is doing is telling us how good are our intensity simulations, which is what we want for neuromodulation. And so this is just a nice data set that ultimately all it's really doing is going to validate our intensity simulations, which is what we want for neuromodulation. You know, neuromodulation, we're gonna, yeah, we care about the temperature, but mostly we're gonna care about what does this intensity look like? What is the shape of the intensity? Where is it in the brain? And looking at this data and looking, you know, sort of angsting over the, you know, the temperature rise is just because it's a good data set, because we've got truth with our MR temperature rise. So that's why I'm spending so much time working on this, because this is a nice validation of beam simulations. Okay, so we also did measurements of attenuation with those same skull bits. Whole bunch of different ones. Now in this case, we split it out so that we made sure that we were more cognizant of the fragment size, because when the fragment size is really small compared to a wavelength, then there could be, you know, other considerations that we weren't really taking into account. So we really wanted to really include only the fragments that had a thickness that was on the order of a wavelength or bigger. We did the same thing, hydrophone with and without the skull fragment. But in this case, now the way we analyzed it was that each of those pulses, we did the Fourier transform. And we know this from the lectures that we've seen previously, is that a pulse like this is short in time. So that means it's going to have frequency components that are rather broad. And if you look down here, if you look at the frequency spectrum, we see that each of these does have frequency components that are rather broad. We looked at it with and without the bone. And so you can see if you look for yellow, for example, with the bone and without the bone, that there's differences. And actually, this is really interesting, because what we see is that the differences are really small at low frequencies, and they get bigger and bigger at high frequencies. Well, this makes sense, right? Because you know there's more attenuation with frequency. And so that just explains why there's a bigger loss at the higher frequencies than there is at the low frequencies.
And each color here is representing a different skull fragment?
Nope, each of the colors here, this is one fragment, and we did these measurements 3 times over with 3 different transducers with 3 different center frequencies. So the yellow one had a center frequency at 2.25 megahertz. The red one was 1 megahertz, and then the blue one is 0.5 megahertz. So even though they had a center frequency that was very specific, because we use these short pulses, they had a bandwidth associated with them. And so there were, you know, a broad frequency components. So by taking the Fourier transform, we were able to look at those broad frequency components and not just be stuck with only just the center frequency. So that meant that for every measurement, we could actually make multiple measurements of what the loss was as a function of frequency. So then we said, okay, these are all our measurements. We're going to include all the measurements where the without bone was within 10 dB of the peak, and the with bone was within 50 dB of the peak. And so like out here, for example, we did not include this because the without bone was below that threshold. So that was our inclusion criteria. What we ended up was filling a grid that looked like this. So this was frequency this way, and then this is Hounsfield units this way. So remember, we had 100 fragments with different Hounsfield units. So we had 100 different lines in here. And then we had, from the data in the prior slide, remember there was 3 different center frequencies for the 3 different transducers, but then there was some broad bandwidth. And so there was multiple measurements for each bone fragment in the frequency component direction. So having made multiple measurements and filling up a grid, then we fit to a 2D function that went this way. So it was sort of an exponential in the Hounsfield unit direction, and then an exponential in the frequency direction. And what we saw is that there was the hot spot over here with higher attenuation, very, very high attenuation over here. This corner is lower Hounsfield units, meaning it's where there's trabecular bones, there's more scattering. It's at higher frequency, And we know that the attenuation is frequency dependent. Higher frequency is going to give you more attenuation. So both those counts, this makes sense that this is the high corner. So we fit it to the plane, and then that was now the attenuation curves that we can use going forward. And of course, we did this for multiple different sets of CT images. Yeah, so just showing you before we move on from this plot, this is just the grid that we filled up, and there are samples. And then this is, if you plot these 3 different lines, so looking at sort of the center frequency across the different samples, then you see the dots are the data points, and then the lines are the fit, where the fit isn't to these particular data points, the fit is to the 2D curve over here. And having fit that, then you can see, well, on these lines, how well did those particular data points fit the curves that we fit from all of the data. And then you can see that they fit reasonably well. And again, we see the sort of relationship that we expected, where it's kind of increasing, and not just linearly, but sort of quadratically with lower Hounsfield units. And then in the frequency is sort of increasing at higher frequency as well. Okay, so that's the data that we have for skull attenuation. You know, I should mention that other people have looked at both speed of sound and skull attenuation and attenuation for bone. And our work is the work that I think has been the most complete in terms of first making measurements, but also making measurements as a function of the imaging parameters. So you can go back to the Fry papers where they made measurements, but they didn't have any imaging because that was before, you know, development of the CT scanner. So they didn't have any imaging correlates. They could just say, well, this is what the speed of sound was as a function of density, which is something they could measure. But here we're really interested in being able to do these beam simulations and being able to find acoustic parameters as a function of, you know, every pixel, every voxel within the volume. So having imaging is really important. There's a number of other people who have looked at this as well. A number of other good researchers. Some of the other methods they've looked at is that they've looked at what are the losses that they've gotten, and then, you know, what are the parameters of the skull that were consistent with those losses. You know, the parameters based the Hounsfield units, or what's the relationship of the parameters on Hounsfield units that were consistent with having made an intensity loss across, intensity loss measurement across the skull. So it's a little bit different than having actually sort of directly made those measurements.
And Kim, is this only important because using a phased array, we can actually correct for different attenuation at different areas of the skull?
Even with your single element transducers, you're still going to want to know what the attenuation is so that you know with a single element transducer, what's the intensity at your focal spot.
Oh, and like how much smearing and how bad it would look.
Yeah, even with your single element transducers.
So if I'm trying to build you know, a new device and I have a single element and it looks bad, you know, it just smears too much because there's differential attenuation, I just basically have to get rid of that element and use a different one. Whereas if I had an array, a phased array, I could vary the intensity across the array to correct.
Yeah, you can vary the phase as well as the intensity to correct for it with a phased array. With a single element, you put up with it, or you move your transducer to another location if possible.
Mm-hmm.
Those are your only options. Yeah. Okay. So far I've talked a lot about CT. CT images are beautiful. They give you beautiful bone images. So there's a nice one there. And I could actually show you, or maybe I did already, some zoom in of CT images where you get beautiful detail of the bone, especially the trabecular bone. And CT is great, you know, for essential tremor patients who are 65 to 85, they can go and get a CT, and there's so much research these days on how to get low dose CT, low radiation dose CT, that it's not really as bad as it used to be in terms of radiation dose. But on the other hand, if you take somebody who's like, you know, young researcher, maybe Keith, for example, and you say, well, I want to do a neuromodulation study on him, it sure would be nice to not have to get a CT. and be able to do something with an MRI, for example. And especially in the future, you know, I think fMRI is going to be a tool that we're definitely going to be wanting to use when we modulate the brain and look at the brain and at the fMRI to see, you know, what's the effect of what we're doing. So we may already have an MR scanner or be, you know, in the MR scanner. So being able to get some of the information from MR could be useful.
Do you go on to show a picture of an MRI? I forget, or are you just kind of discussing it?
Thanks for asking that question. So here's a nice example where we have a CT picture of a skull over here, and these are human skulls in vivo. This is an alive person. And then we have two MRI pictures. So the one of the most on the right is the most similar to conventional MRI. In conventional MRI, we're looking at the water protons, and the fact that the skull doesn't really have a lot of, I mean, it does, it has a lot of water protons, but they're fewer than in the soft tissue. And so the skull looks dark on conventional MRI. Now on this set of images, what we've done is we've just used a really, really short echo time so that we can get a little bit more of the signal from the water that is in the bone. And then we manipulated the image, so we just kind of inverted it. And then you get an image that looks like the one in the middle. And now you can see that the contrast is looking very similar to what we see with the CT. You know, the resolution isn't quite as good yet, and we don't really know all the details of how to relate our MR parameters to our CT, to our acoustic parameters, but it's getting there. We're doing some simulations to try to see, can we do the phase aberration correction just as well with the MR that looks like this as we could with the CT? You know, the resolution isn't as good, but how important is resolution for that task? So we're trying to answer those questions, and we think that at this point, MRI is behind CT, but it's still getting better.
Yeah, I'll harp on the point really quick. If you're trying to make a product or a usable thing, you really don't want to pay, first of all, for a CT and an MRI. There's time and like huge amounts of cost associated with that. And Kim mentioned this, but with an MRI, you're getting a really detailed image of the brain, which these days the tools to map the regions of the brain are very quick and very easy. And a CT gives you none of this. So what Kim's work is trying to do, you know, relating CT and MRI stick or Hounsfield units, it's really important.
Yeah, so there's going to be 3 roles to the MRI. The first one is for the bone when we're good enough. The second one is for targeting. So that's what Keith was saying, is that the MRI gives us all that soft tissue contrast in the brain to target the neural structures that you don't see on the CT. So on the CT, you can maybe window in and you can maybe see where the sulci are, you know, to be able to see the CSF versus the gray matter, white matter, but you're not going to be able to see the details of the neural structures that you can see with an MRI. And then lastly, it would be an fMRI type readout. Okay, anything else on that part, Keith?
No, I don't think so. Well,
Okay.
actually, are we gonna, I forget, in later lectures, it'd be cool if we taught students the typical methods for actually mapping the brain as it relates. I think we do. If we don't, we should. You can think about it.
All right, we'll talk offline.
Yeah.
All right, so where are we? We've talked now about assigning acoustic parameters. So there's one topic else I want to talk about. I think this lecture is going a bit longer than I thought it was, but I'm going to go ahead and try to squeeze in here with the phase aberration correction terms. And then the rest of it, we'll just wait and hold off on another lecture. Okay, phase aberration correction terms. So the basic idea, I think we've talked about this a number of times. You have some number of elements, and as they traverse the skull, they're going to accumulate phase, which is because the skull has a different acoustic velocity, and it has each of those has a different path length, and they're going to have different phase of the focus. So the simple ray tracing is that you define a ray from your transducer element to the focus. You figure out, you know, every voxel it goes through and the speed of sound through that voxel. So you're calculating here the different speed of sound through each of those voxels that goes through the skull, and it's accumulating phase through each of those voxels. And now each of these, because they've gone through different path lengths of different bone with different acoustic parameters, different velocities. Now they're going to have different phases. You take each of those phases, apply the negative to the transducer elements, and now you've done your phase aberration correction. So that's the simple ray tracing.
I just want to remind everyone when Kim says accumulated, even so, if you're accumulating an entire cycle, you're now back in phase with the original point. So it's sort of like accumulation, but it's sort of like a phase that just wraps back around. Is that right? Am I saying that right? Because everyone was confused about that.
You're right. I mean, if you accumulate 2 pi a phase, then it's going to be back in phase again. That's exactly.
It's sort of accumulating, but it's sort of just going in a little loopy.
It's just that it's unlikely to be exactly 2 pi a phase. It's going to be all spread out, and so they're going to be all different phases.
Right. Yeah,
okay.
Yeah, that's just it can happen though.
So that's the simple ray tracing, and that doesn't take into account reflection, refraction. There's lots of things it doesn't do. But let's just ask the question: how well does that do in terms of fixing up your focal spot? And so Steve did this, and I'm going to tell you a little bit about his work. This is Steve Leung in my lab. He actually used the Insightec transducer, so there was 1,000 elements. So that's why I pictured it here with more elements. So he took 3 ex vivo skulls, and he put them in a head holder, and then he immersed that in the Insightec transducer. So you can see now with the skull is now immersed in the water bath. 3 skulls, they did have CT images, and he also got MR. So at another year, I'll be able to tell you the results with the MR. But right now, I'm just going to tell you about the results with the CT. These are 3 skulls that are very different. They have different thicknesses, definitely different shapes, and different densities. All right, so on every pixel, he took those Hounsfield units, and then he assigned it an acoustic velocity. And then the curve they used was the black one right here, which is very, very similar to the curve that Taylor Webb found in the work that I showed you above, where he was taking his little bone fragments and measuring the speed of sound. So those are very, very similar. Okay, so then what Steve did was he had his skull in there in the transducer, and he used a hydrophone. So yay, we talked about hydrophones and how those work, and the fact that you can scan the hydrophone around in order to get a picture of the focal spot. What he did was initially he set it up so that it was right at the focus, and then he turned each of these elements on individually. When he turned one element on and he measured with the hydrophone, he was able to measure the phase of that pulse that hit the hydrophone. And then for the next element, he did the same thing. He turned it on, he measured the phase. So he could use each of those phases as measured by the hydrophone to correct for the skull phase aberration correction. And that's the best you can get in terms of the correction. That is our gold standard for correction, hydrophone correction. Of course, this only works with an ex vivo skull. All right, so he looked at 5 methods for phase aberration correction. The first one was no correction, and the last one was using his hydrophone. So that's going to be the best of all possible worlds. And then what he did was he said, well, let's look at the focus right there, and we'll be looking at the intensity of the focus. Now, before I move on, I got to tell you about the other 3 methods. So the first one was the straight ray tracing. It's basically what I told you in the last slide. For every element, you figure out the beam path that it's going to as it goes towards the transducer, and then you correct for the phase of the beam, and then you just apply that on the transducer. All right, so he also used the Insightec system, so they have a workstation. He was able to plug into that what his CT scan was, and used the phase aberration correction terms that came out of that. Now, we don't know the details of how that works. It's obviously different than the straight ray tracing. And what you see is the focal spot was displaced, which isn't good necessarily, but it was definitely more in focus at the displaced focal spot. And that's kind of interesting because the Insightec system is using MR temperature maps in order to get some verification of the focal spot. So for them, it doesn't really matter that the focus is off center, because they can measure it's off center and they can use their electronic focusing and steering to push it to the correct location. So it actually works because they've got that MR feedback. If you're in the situation where, you have a skull and you're outside the MR and you're not going to get a temperature rise and you don't have any imaging feedback, you don't necessarily want it to not be at the right location. So it is something to be concerned about. And then the last method here is what's called HAZ. This is using a hybrid angular spectrum full beam simulation that I haven't told you about yet. But that's just another method for sort of for each element calculating what the phases are through the skull. It's not the simple ray tracing, it uses the full beam simulation. And I can tell you about that right now, but that's just his comparison. Okay, so we got 5 methods and we're gonna say how well are we gonna do for correcting for those phases. And just looking at one skull, skull C, what we see is in the case where there's no correction at all, that the intensity is pretty low right there compared to the hydrophone. So this is all normalized to the intensity we got at the peak intensity with the hydrophone correction. So it's only about 10% if you don't use any correction. With a straight ray tracing, we're up at about 50%. With the Insight system, not good there, but if you actually measured it at the focus, it's much better. And then with the HAZ method, it's better. And so this is now if you're looking at the focus, the Insight system did much better. Now this is just averaging across the 3 skulls. What we see is that, I don't know, maybe about 30% in the case where there's no correction. And then if you use the straight ray tracing, you can get about 60% of your intensity back again. And that's compared to the best you can do with the hydrophone. And then using the other methods, you can do it maybe a bit better. The HAZ method, you can get up to maybe about 80%. So this gives you an idea of the importance of phase aberration correction. It tells you you can get a lot of intensity back just with a straight ray tracing. It tells you with the acoustic properties that we're assuming, what can you do for getting the intensity back, and that you can get, what I'm showing you is how much you can get back. It also shows you that there's still more to be done. We'd like our intensity to be the same as when you use the hydrophone correction, but there's still some work to be done there. Yeah, so one of the problems is that variation in the acoustic velocity as a function of Hounsfield units that I mentioned before. And it's this variation that's causing all of these methods to be lower than the hydrophone method. So that's just something still to be worked on. All right, I'm going to stop soon after we finish talking. We talked about what the key elements are. You model your transducer. On every voxel, you have to assign parameters. If it's an array, you can do the phase correction, and then you propagate the wavefronts, calculate your intensity at the focus, and then calculate the temperature from there. So we've talked about some of the elements. We haven't yet talked about propagating the wavefront and how to do that in simulation. So I'm going to save that for a later lecture. All right, Keith, you got any questions before we sign off?
God, we've gone on so long with this lecture, but I will quickly say like a hats off to you and Steve for that model improvement. I mean, 15 to 20% improvement and not having to do thermal shifts in the brain to actually find the focus point. It's impressive. We don't want to have to heat up the brain to know what's going on. So I hope that your work and Steve's work continues to improve upon that measurement.
Yeah, I'm a firm believer in modeling and being as accurate as possible with the modeling, because if we're going to use this as a neuroscience tool, we're going to have to do really accurate modeling in order to try to get reproducible intensities across different people's skulls. It's just going to be critical.
And I'm sure as MRIs and imaging tools get better, so will your simulations. I guess we'll just see where that goes. Until next time.
Yeah, until next time. All right, good. Thanks, Keith.
Thanks, Kim. Bye.